Research and Programs Department, and I will be the moderator for this session. Maybe a few housekeeping. For issues of CPD, kindly email us at knhcpd@gmail.com or at cpdpharmacy@gmail.com. Concerning pharmacists and pharmaceutical technologists, we got word that for now we are not able to upload the CPDs on the PPB wall. So because of that, then it means we, as pharmacists and pharmaceutical technologies, you need to self-report so that you can be able to get the CPD points. So how it goes about is on the Pharmacy and Poisons Board portal, there's a section for self-reporting. Kindly click on the self-reporting icon to be able to make your CPD claim. On that self-reporting icon, there are several fields that you need to fill. And once you fill them out, there's an area where they'll ask you for evidence. So the KNH, the Research and Programs Department will be emailing your attendance lists, and that is what you will use to upload on the self-reporting as the evidence for CPD. I hope that is a bit clearer. For now, we are not able to upload the webinars that we are holding on the PPB portal. However, once you self-report, you'll be able to get your CPD points without any challenges. And in case of any issues, please feel free to let us know and we will try and address them. Right now, I'd like to give the floor to Dr. Esther Miner and Professor Christian Dels. They'll introduce themselves and they can begin the session. Thank you very much.
Good afternoon. I'm Esther Miner, Department of Biochemistry, and I think I will start us off by giving an introduction into genomics, and more specifically, I'll be talking about the African genome, the reasons why we need to study it, what are the advances in technology that we are experiencing at the moment, which actually strengthen our fight or our cause towards understanding our genome, and what is the end story. And some of the work that the group that I work with is involved in. So I will try to share. I don't know, do I have the rights to share?
Yes, you do.
Okay. Because I cannot see the sharing. I'm a bit lost. Oh, share. Okay. Thank you, Simi. So. So I'll just start. Okay, thank you very much. So again, like I said, I'll be talking about the African genome, and more so for this session, it will be quite introductory and actually just trying to drive the issue or the agenda of normalizing this narrative in the African scientific fraternity. So I've actually termed my talk The Long Walk Home, case in point being Africa is the cradle of mankind. So yeah, so I will begin. So a genome is a complete set of genetic information of an organism. It actually includes all your genes and the proteins, which actually subsequently give us the functions therein. So the bringing together of all these moieties actually are what give us a genome. So we'll look at that and why we need to study it, and some evidences as to why the African genome needs to be given special attention, some evidences, and then we'll take it from there. So this is a quotation by Charles Rotimi, the gentleman from NIH who started off the H3 project or consortium, which kicked off the studying of the African genome, where he says that there are things in the human genome that we cannot study anywhere else, but on the African context. So he goes on to strengthen my argument that Africa is the cradle of mankind. Therein lies all the basic and the originals of genomic information that we so seek. So why do we need to study the genomic sphere of Africa? So genome research actually is search for variation. That is how I see it, and other researchers. Roughly, we do know that at least 99.9% of our genomes across populations are quite similar. But what remains different, or the difference therein, is what is important. This is what we are seeking after, because it will give us answers that lead us to actually determine what causes our diseases, both communicable and non-communicable. I'm a non-communicable disease researcher, specifically cancer. So finding those differences will go a long way to enable us determine or interrogate the billions of nucleotides in our genome towards that end. The lack of African genetic material, so we do not have biobanks because we do not have those genetic materials. This constitutes significantly an obstacle, or poses as an obstacle to our understanding of how our bodies and diseases function. Case in point is the narrative of cancers in Africa, and I'm sure many of you have encountered patients who actually show very poor response to chemotherapeutic agents. We do not understand the mechanism of disease because they are not actually working together with what is in data at the moment from the Caucasians, which is the other population. Yeah. So Europe actually constitutes 16% of global population. But if you look at the genomic information, or the DNA information that is out there, it constitutes from Europe. It is quite, Africans actually are actually presenting some dismal presentation, because we only present 2%. That is how little we participate in terms of these genetic studies, and that is to our detriment. Means that our populations are significantly and severely underrepresented. And so in the event that we wanted to find these differences that I so daily talk about, issues of comparative studies of genotype, phenotype correlation studies are not possible at all. So it is important to actually push forward the issue of understanding the human genome, so that we'll be able to do comparative studies of our diverse population and understand the genetic basis of the phenotype, adaptation, and complex diseases that we see in all our populations. So Africa actually hosts the majority of human genetic diversity, which we'll see shortly. Obviously holds the clues to human origin, is the cradle of mankind, and unfortunately carrying the heaviest disease burden because of our diversity. Yet genomics has so far focused on our European counterparts, but now Africa and other populations are looking forward or hoping with the advancement of research and techniques and technology to reap the benefits as well. So globally, human populations show structured genetic diversity, and this is as a result of geographical dispersion, selection index, what pressures are put on us, and so it even affects matters of drifting. These variations subsequently will provide insights into the evolutionary processes that shape both human adaptation and variation in disease susceptibility, not only to the Africans, but to the human race, so to speak. There are two projects that have greatly enhanced our understanding of genetic variation, and they're sort of early days. the Hapman and the 1,000 Genome projects, which have been quite advanced and enhanced our understanding. But the characterization of African population remains limited or wanting, so to speak. So this is the first, I've selected work by this scientist, Gardasini. She's done a lot of work, and I've started with some of the work that she's done nearer to home, which is work that they did in Uganda, looking at the population history and genomic discovery. That work was done in Uganda. And it really was shocking. This was work done in 2019. The first report was that more than a quarter of the genetic variation that were found among the people of 25 Ugandan villages has never been recorded previously. So that is just Uganda. We're talking about 25 little villages. And so in the grand picture, in the grand scale of issues, if you were to think of Africa in its entirety and the diversity of the people therein, then one must then be able to project and see that we're actually quite wanting in this. And that is why, again, I would say that this narrative has to be pushed forward. Then after that, Gradasini et al., or actually it was not after, these are two things that that group has done. They had actually started looking at the Sub-Saharan Africa, looking at the genome variation under the African Genome Variation Project, which is an international collaborative effort that has actually expanded the efforts to systematically study and assess diversity among 1,481 individuals from 18 ethno-linguistic groups. And you'll be happy to know that Kenya was involved in this one. We had the Kikuyus and the Kalenjins were actually captured. It's just sad that it is not a Kenyan initiative or an advancement as we see in Uganda, because the Ugandans have actually taken the first step to start studying their genome. So actually, they actually got some very interesting results, which I will share shortly. So they used an analysis from all this diverse work. So out of the 1480 individuals, they used 320 to do a whole genome sequencing analysis, which is quite a lot, using the next generation sequencer. So you can imagine to do the whole genome, it was quite impressive. Using the ADMIXTURE analysis, which is one that shows how the substructures within populations actually interact, thereby giving us diversity. How that is what this statistic in the context of Eurasia and the African context, they were able to then show us what is the output of the same. So I will share so that you have an appreciation. You can see that in this presentation from the ADMIXTURE analysis, you can see that the Europe comes in as a block, Asia as a block, the Americas, and then Africa is actually subdivided. When you look at North Africa, it has substructures. These are all genetic diversities, the variants that were reported from this. That should then tell you the amount of data that is wanting, the variability therein that will go a long way to advise us in issues, disease, in issues, for example, in my area, in issues cancers, so that as I ask questions about prostate cancer and breast cancer, then I know that there's a lot of variation, even in my own population. So I thought that was quite interesting. So as a summary, they actually presented 2.2 million modest variants that show differentiation among the sub-Saharan Africans. They did report 29.8 million SNPs, single nucleotide polymorphisms, that were identified from Ethiopia. And in Ethiopia, there are some substructures of the Oromo and the other small tribes therein, the Zulu, the Baganda. So that was work done on the whole genome sequence by the AGVP group. So I hope you now have that understanding and appreciation of the diversity that we have that has not really been tapped in, and that we need to harness our resources and our collaborative efforts to that end. So to conclude the AGVP work, I would then say that African populations are characterized by greater level of genetic diversity, extensive population substructures, as we've seen from the admixture analysis, and so less linkage disequilibrium compared to the non-African populations. Africans also possess a number of genetic adaptations that have evolved in response to diverse climates and diets, the nutrition, because we're very diverse as people of many colors and many traditions and many things, as well as exposure to infectious diseases, which you're obviously very well aware of and familiar. So at least this AGVP result from 2014, 2019 actually should strengthen our wanting to delve into the African genome the more. So in terms of the technologies and the advancements therein, because this is where our salvation comes from, there's been unprecedented advances in molecular and cellular biological tools. For example, the cost of sequencing. There are times when, I remember up to 10 years ago, sequencing was something that could not be discussed in Africa, but up to yesterday, you can actually sequence a primer pair for 1,000 shillings. So you can even fund some of your sequencing work. And so those drivers of innovation in chemistry, optics, fluidics, computational bioinformatic tools, driving the costings of these technologies or biological tools to an affordable level should strengthen us the more to want to get involved in this. So that has actually been very important. And indeed, we're moving from the dideoxy sequencing to now the next generation sequencing, where we can now look and interrogate whole genomes in their entirety at prices which we can now term as affordable. So we're in the multi-omics era, which is quite interesting, and I would probably say we haven't heard of others. We are working, and when I say we, I work in a group of some other researchers from the University of Nairobi and Manchester University to use multi-omic approach to ask specific questions about specific manifestations, and specifically we are looking at prostate cancer. And so we have sections where we are doing the genomics, proteomics, which is looking at structure and function of proteins, transcriptomics, which deals with matters RNA. So we're interested in gene expression and the sequence of the transcriptome. Metabolomics, where we assay for metabolites that come from a situation or a disease, and looking at how that affects that specific manifestation or pathophysiology that we are observing. You know the roles of microbiomes in terms of, for example, in the gut and how the immune system is regulated by that. So basically, we are doing a multi-omic approach towards prostate cancer as our starter point. So towards that end, we will use some statistical analysis, which actually has factors or linker factors that actually give us a weighting to each of the omic towards how much it builds towards a specific manifestation. And like I said, in my area of prostate cancer, we'll be looking at hypoxia that is created in cancerous situations, and look at the metabolites therein and see how much they actually impact on the disease severity or progression, and why we see abnormal, and by abnormal meaning that it deviates from what is in what is published data. So in terms of talking about multi-omic approach, this is what would be able to ascertain, be able to carry out patient stratification, predictions of therapeutic responses, personalized treatment, most important, the therapeutic monitoring, going towards prognosis. So this is bringing together of those omics. So what are the gains from these advances? All these developments define this, all the knowledge of us wanting to challenge the African genome, and then also being supported by advances in tools and what the molecular biology techniques. We should be able to, we are actually in a unique and opportune time where we can seek to understand our African genome. We can now envision obtaining easy access to the complete sequence of 3 billion nucleotides. We can interrogate them further and do our mechanism of action results, and even trying to understand signaling pathways of specific disease, subsequently leading us to genotype-phenotype correlation analysis between our population and populations out there. This knowledge that we'll gain from such a venture will have a major impact on healthcare, disease progression or prevention or management, to also raise challenging issues regarding rational issues and ethical implications, which are also very important. So what are the ongoing things or research work in Africa? And I've actually singled out two major consortiums. The one that was founded by Charles Rotimi, which is a H3 Africa. And you can see it's actually with collaboration with Wellcome Trust, they actually have quite a diverse sort of like collaborative effort across Africa. And this is with a view of covering the whole of Africa, to actually look for the diversity in our genome in the African, and actually creating databases from where we can pull out our genomic information and be able to do subsequent work therein. So H3 Africa is an initiative that consists of 51 African projects that actually deal with both communicable and non-communicable diseases in Africa. What really impresses me about H3 is how it empowers the African researcher in genome science by enhancing or strengthening collaborative efforts, and again, the facilitation of the fundamental research work is actually led by African scientists for the African people. So H3 Africa has gone a long way in issues of creating or strengthening capacity, which is much needed in our background. The other one is 54 Gene, an initiative which is focusing on all the 54 countries in Africa. It's Nigerian based, and it was started by Dr. Bong. Basically, they are actually dealing with drug creation or drug discovery. And this actually is very important because we have the H3 Africa doing the genetic diversity studies, trying to understand the African genome. And then we have this other arm of 54 Gene trying to do drug creation. So, and both of these situations run concurrently, which is exactly what Africa needs now. So what are some of the challenges? The challenges are just the usual challenges that you would have. Of course, there will be some that are a bit odd because of the kind of data that one is dealing with. For example, discovery of incidental findings unrelated to disease in question. Patient awareness in our population is quite difficult. Just the usual. And you will note that I have not mentioned finance. which is actually a global issue. It's an issue that affects all researchers globally. Of importance to me and fellow researchers is who then owns the data. That is a question that we then must ask, because we still are resource constrained in terms of infrastructure. And so we rely heavily on our collaborators to do the slightly more complicated analysis out there. And so we send out our samples. So this is some discussion that has to be ongoing so that we know who owns the data therein. There definitely will be some good opportunities. I'm sure you've seen that in terms of precision medicine, where we're able to personalize treatment. That will come from understanding of the genes and be able to use that information to actually tailor-make treatment. A good example is what was done 20 years ago by Golub et al. Matters leukemia, where Golub et al. Came up with a subset of 6 genes that could actually then break down leukemias and be able to very quickly determine the exact leukemia that a patient is presenting, subsequently quickening the treatment protocol, and it is quite precise. And that is where we are actually looking to go. And in as much as it is a lot and it's quite demanding, I don't think it's insurmountable. I would not want to finish this work without talking about novel biomarkers. That has actually been my absolute interest in matters cancers in Africa, because I'm very sort of pushed by matters of how the diseases are progressing, the severity, the prognosis. If we're able to identify subsets of markers that are unique to our populations, for example, in breast cancer, will go a long way in understanding these diseases and being able to understand the molecular biology therein. Other issues are issues of costs. Can we come up with cost-effective designs for carrying out genome-wide association studies in Africa? Because you want to then, once you've pulled out the biomarkers, associate them with a specific phenotype, and so have an ending to a story, thereby leading you to a way of sorting out or mitigating the situation. Of course, the other good things that will come out of all of this is a collaboration across the regions, strengthening research capacity. We envisage data from these projects will provide a global resource for researchers, as well as facilitate genetic studies in Africa. Of course, research infrastructure overall improvement in disease management. So I would like to acknowledge the team that I work with. These are the people involved in the multi-omics team, and hoping that the next meeting or the next invite I get from this background, we will be having some very interesting results from our prostate cancer. So there's Dr. Ezekiel Mecha, Dr. David Wedge, Professor Peter Mungai, Dr. Esther Kanduma, Dr. Martin Mlinge, and Professor Robert Bristol. And for me, Esther Maina, thank you very much. And I would like to welcome any questions or discussions, comments. Yes, thank you.
Thank you very much, Dr. Esther Maina, for that lovely presentation and bringing up the issues concerning the genomics, especially in sub-Saharan Africa and in Kenya in particular. I'd like to give the opportunity to Professor Christian Dels to give his to introduce himself and give his presentation. Then we can take all the questions together at the end.
Okay, so that makes a lot of sense. Thanks very much, Nancy, and thanks, Esther. This was a brilliant presentation. I'm a little bit anxious now how I can top this with my presentation, but you gave me a really good lead in and presented a very strong case why it's so important. What I would like to do is to look at the other end of the spectrum, obviously a little bit more into the environmental roles and on top of the genetic background. So what I'm trying to do is just to share my screen with you. Let's see if this works. And we'll take it from there. So, right, so I'm based at the University of Glasgow in the UK. I do not have any disclosures for this talk. And what I have as a title, obviously, is the interaction between genes and the environment. But you will see that along the talk, we will focus more on environment than on genes. And that's actually good because Esther has really covered the genetic background quite well already. So there's no need to go too much into this. But there is a lot of studies that really look into environmental exposures together with the genetic background. And let's explore this a little bit. And one of the areas that I'm really interested in is hypertension. And hypertension, as you all know, is the major risk factor for any cardiovascular diseases worldwide. And it is certainly with cardiovascular disease being so important as a major cause for morbidity and mortality, it's certainly worth to look into hypertension. But when you look into hypertension, you see that it's not very common in the younger people, but it is, of course, very common in the older people. And this is already giving you some evidence that there could be an interaction between the genetic predisposition and environmental factors. Because why on earth should this prevalence of the disease increase with age if there is not an interaction between what we all bring with us as genetic background here and then say 50 or 60 years of exposure to all sorts of environmental factors that make it then more and more likely to develop hypertension. And of course, you also know that if you look at hypertension, there may be people who have high blood pressure exclusively because of genetic causes. So that would be people, for example, who have monogenic syndromes of high blood pressure. And in these people, it would be quite reasonable to say that their hypertension is exclusively driven by genetic factors. But you may have the other extreme where you can probably, by a combination of all these bad things here, also have high blood pressure without a particular genetic predisposition. So there could also be the other extreme that only environmental factors drive hypertension. In reality, of course, in essential hypertension, we will always have a combination of genetic factors and environmental factors that act together here. So I often use this schematic here when it comes to any sort of cardiovascular diseases, because we need to make ourselves aware that obviously any sort of cardiovascular disease, such as congestive heart failure or end-stage renal disease, they do not usually develop overnight. They develop from early stages, functional changes, more and more structural changes, and then established disease. And if we again look at the role of the genome here, the genome is very early, of course, because that is something that does not change throughout life. But what clearly changes throughout life is the expression of genes, and then, of course, the expression of proteins and metabolites and all the other things that we've already heard that will then change. And these would be certainly the factors that could be influenced by environmental factors. So let's look into a few of these things here. And one is in this paper, the authors have really looked into the environmental interactions according to a number of genetic factors. And what is very helpful here is this first table. And obviously, I don't want to go through all the details here, but just the principle is quite interesting, just looking at the environmental exposures. And you see three different hypertension guidelines here, the European guidelines, the American guidelines, and the Japanese guidelines. And they just look into these guidelines and say, what are the recommendations for environmental factors, such as diet and other factors that are across these guidelines. And you see that these are already quite different. In terms of salt intake, for example, there are differences, or slight differences between the European, the American, and the Japanese guidelines. In terms of physical exercise, in terms of what is a normal weight or a normal BMI, there are differences. And that, of course, just shows what we all know, that these people are different between America, between Europe, and between Japan and other parts in the world. And of course, not only in terms of the genetic background, but also in terms of their environmental habits. And accordingly, we have different recommendations here. So that is one factor that already makes it difficult if we talk about gene-environment interactions, that obviously not only the genome is different, but also the environment is different across the world. So if you look at some specific factors in diet, for example, that is something that plays a role across the world. We have very recently, together with the Lancet Commission on Hypertension group, the CDC, and the World Hypertension League, published a series of papers from young investigators in low and middle income countries. For example, this one here in Pakistan, or another paper here from Russia, where we looked at trans fat or trans fatty acid intake. And you see that just randomly taking this from 2 different countries or regions in the world, it is a problem everywhere. So that is something where clearly low and middle income countries play as much of a role as any other countries in the world to drive the research here and to make something healthier, so to make sure that our populations have a much healthier diet, because that is obviously one thing that we can influence. There is the other issue really about salt intake, and that is something that also within our network of excellence, we should, or center of excellence, we should really look into this in a bit more detail, because salt reduction is something that across the world lowers blood pressure, reduces the risk of cardiovascular diseases. But the strategies are, of course, completely different depending on which country you are, because of the dietary habits being so different. And this is a paper from Mohsen Ibrahim in Egypt, where he summarized hypertension in developing countries and has also salt reduction as one of the most important aims here. But I know from their own work, but also from work from others, that of course diets across, for example, just the African continent, but also across the whole world is of course completely different. And there may be different sources of salt in different countries, and that needs to be addressed very specifically. And we know this from any studies that we do into dietary habits. So there is certainly something we can modify, but it needs to be targeted and it needs to be investigated much further. And we can't just, as in the genetic world, we can't just translate things that we have done in Europe or in the US to other countries or continents. And it's particularly bad, of course, so that's a picture of a German diet. If you combine the trans fats and the salt, then probably you have something really bad, and that would be something that can be quite easily fixed. However, if we talk about healthy diet, and healthy diet can be defined in a number of different ways here, then if you look at interactions between blood pressure, healthy diet, and the genome or certain genetic factors, you see that across these different regions of the world, different genes are associated or interact with healthy diet and blood pressure. And these are all very small studies, but you see already from this small table here from this paper that there is not a uniform signal. So it seems to be something that is, again, country-specific or area-specific or community-specific, but not something that we can easily say that's true across the world. And it's certainly also an under-researched area. And this is deliberately small so that you don't look at the details here, because this shows the interaction between genes and alcohol and blood pressure. And you see a large number of genes here that have been identified. But again, different genes across different studies, across different parts of the world, not a very consistent picture here. And again, rather small studies that have been done here. So the summary of this paper was that a sufficient amount of evidence has not yet been accumulated, and the results of genetic factors often are different across the studies. And I think that's very important. We all know, of course, that there is an interaction between, say, blood pressure, alcohol intake, and the genetic background, but we don't have robust evidence at the moment to say in this population we can pinpoint it to a specific gene. And I think that's, again, something where with the sequencing data that Esther has already mentioned, that will give us a much clearer picture, and hopefully that could be also some of the research that we can do within our Center of Excellence. Now, the other thing is, and we've heard this also before, that obviously doing the wet lab work is one thing, but then to analyze such data can be very complex and very computationally demanding. And also when it just comes to the very basic concept, because if you take this environmental factor and the genetic factor influencing this, you could have different scenarios. So one scenario would be that you have a number of environmental factors. They all influence some sort of intermediate phenotype, and this leads to the outcome in the longer term. So that could be, for example, vascular dysfunction, endothelial dysfunction, and that could be a heart attack. And we do not know where do the genes interact with all of this. This could be the poor diet and lack of exercise. Would the genetic factors interact here at this point in the development of disease, or would genetic factors just interact in a different way, for example, in the way how one specific environmental factor influences the outcome? We don't know this in every case, but different models, different concepts would require different statistical methods to analyze this. And because we don't know so much about this area, there is a bit of an issue which kind of statistical methods to use to analyze such complex data in the best possible way. The next slide actually just gives you an illustration of this, and I really don't want to go into the details of what exactly it shows, but you see already that whatever these pictures show, these are QQ plots, whatever these pictures show, they are different here. And they are different for an interaction here, looking at different cardiovascular phenotypes, such as type 2 diabetes, obesity, or hypertension, or coronary heart disease, and then using different tests to analyze these interactions with environmental factors, and also using different assumptions, how strong the environmental factors per each variable, how big will be the effect per variable on the interaction. And these are real life data, but you see that depending on the statistical tests that you apply, you will get different results, probably best illustrated here by the p-values that are different across the different tests that can be applied. And that is related to the lack of our knowledge how exactly environmental factors interact with the genetic factors to drive a certain outcome. So there's a lot of things that we need to consider in such research. So there is certainly tools available, but we need to look into specific cohorts. The tools, the guidelines are available. It is possible to analyze such data, to look at interactions between genetic factors and environmental factors, but we need to be really careful to apply the right tools to get the right answers. So if we look at how this really could interact, then I think this paper here and another paper that I will show you in a few slides gives you a little bit of an idea how straightforward this could actually be. These are data actually from the UK Biobank here. And what you see is that when you look at cardiovascular disease overall, or if you look at myocardial infarction or stroke, you see exactly what Esther has shown us already, that there is, in terms of genetic risk here, for example, of course, there are people who have a higher genetic risk of cardiovascular disease and people who have a lower risk of cardiovascular disease, and those with a lower risk have fewer events than those with the higher risk. And the same is true for the other conditions. But you see pretty much the same when you don't look at genetic risk, but if you just look at lifestyle risk. And that could be factors like obesity, that could be factors like diet, or how much exercise people do. So again, you can predict the rate of development of such diseases, also based on how physically active people are or what diet they are. And if you take the two together, you see, for example, for systolic blood pressure here, that we always have three groups here, and the three groups are because of the three different lifestyle factors. So this is the cluster here with the lowest genetic risk. Here you have the cluster with the intermediate genetic risk, and here you have the highest genetic risk. But within each of these genetic factors, the environment plays a role, because those with the best lifestyle have the best outcome, and those with the worst lifestyle on top of their genetic factors have the worst outcome. Of course, that's good news. So even if you carry really bad genes, if I may say so, you can still, with a good lifestyle, counterbalance some of this. Whereas, of course, even the best genetic background does not protect you if you have a really bad lifestyle. But it shows just how powerful the environmental factors are on top of the genetic background. Because if you don't take the environmental factors into account, you may not even be able to see any of the genetic associations here. And I think that is something that people who did genome-wide association studies have criticized, of course, now when they look back to the work that they have done, that they very much focused on the outcomes and the genetic factors, but have not taken the environmental factors as much into account as they should, because some of the negative or neutral genome-wide association studies were probably because we didn't look well enough into the interaction with the environment. So what you saw here for systolic blood pressure is also true for diastolic blood pressure, maybe even stronger, the association here. So really important to see that on top of the genetic risk, very clearly the environmental risk is also there. So given the importance of population-wide lifestyle modification, the use of genetic information for risk stratification merits careful evaluation before it is routinely implemented in clinical practice. And I think that's really, really important. You know, all these commercial people who sell you some sort of genetic risk score or ancestry score and other things, that is all very important, but don't forget that on top of your genetic risk score, or on top of your genetic risk, you always have a huge environmental risk driven by your lifestyle. And I think that's something we can quite easily tweak and fix, whereas of course it's much more difficult to change our genetic background. So moving a little bit away from the genetic background, this is another very topical area, and again, across the world for very different reasons. It may be diesel exhausts in some countries, it may be smoke from fireplaces in other countries, it may be industrial pollution in some places, it could be pollution coming from other sources, again, in other places. But air pollution is something that is really important in terms of high blood pressure. And this is a study that was done in Taiwan, and with stronger environmental regulations, the areas that had high levels of pollution have changed over the years. And you see this just magnified here. So if you go back to 2000, you have a lot of the red color here. So a lot of areas in cities where there was a lot of pollution. And you see that over the years, the red color is disappearing more and more. So the air is overall getting cleaner here. And this is a really good study because it's not only cross-sectional in 1 year, it really shows a development, and it also shows a development of the prevalence of hypertension. And what the authors have shown very nicely is that the higher the pollution was or still is, the higher the risk, the hazard ratio of developing hypertension. But if you lower the pollution rate, you can eliminate some of the hypertension in this area. So very clearly, the genetic background of these Taiwanese people has not changed over these few years, but their environment has changed. And this has very nicely changed their prevalence of hypertension. So it shows again how powerful environmental factors can be. So the beneficial effects of air quality improvement in hypertension is very clear from this study, and it shows again how important it is to improve, for example, things like pollution, air pollution, in terms of cardiovascular health. So I would finally, just come back to another theme that Esther has already mentioned, and that's the microbiome. And that is certainly very trendy at the moment to look at bacteria in all parts of our body. But there is, of course, a very good reason for doing so. Not only that, there's a number of papers published, and these papers address all different areas, including the autonomic nervous system, but it's, I think, something that we can quite easily understand if we look at specific metabolites and specific, in that case, for example, these short-chain fatty acids. They are produced in the gut by certain bacteria, and they are produced because of dietary fiber. So if you have these bacteria, if you have enough dietary fiber, then you produce enough of these short-chain fatty acids, like the propionate here, for example. And all of these short-chain fatty acids, they really play an important role in cardiovascular health, whether it's in the heart itself, in the kidney, in the vasculature, in the brain, in the immune system, anywhere. The problem is that our diet does not include enough fiber, and that our microbiome changes, and we don't even have enough of these bacteria anymore. So the amount of short-chain fatty acids that we produce and then goes into the bloodstream can be much lower. And this is something that directly shows me that the microbiome indeed plays a role, because I can understand this bacteria fiber breaking it down to these short-chain fatty acids, and they immediately have an effect here. And if I change anything here, then obviously there will be adverse effects. And you see these effects here. Again, this is what I mentioned, that they play a role everywhere, but I can show you some data here from studies where people have given these short-chain fatty acids like the C3 here, the propionate. And you see that, for example, when it comes to blood pressure, this is a model where angiotensin II increased the blood pressure, but the animals that were treated with the C3 short-chain fatty acid had less high blood pressure than those who were not treated with this. And there's a lot of other beneficial effects for which we don't need to go in detail. You see diastolic blood pressure showed exactly the same. You see that arrhythmias in this model have changed, so the C3 fatty acids do not only affect blood pressure, but they also reduce cardiac arrhythmias, and they affect things like collagen synthesis or fibrosis, so that you have a lot of this in the angiotensin II treated animals, but the animals who have angiotensin II and the short-chain fatty acid have less of the fibrosis. And this is just another picture here showing plaque formation. Again, a very beneficial effect here compared to this huge plaque in the other animals. So again, all of this and the conclusion that they made here obviously is that this could be an affordable intervention to give an augmentation of this C3 short-chain fatty acid. But for me, the conclusion is much more that we should have a healthy diet, because then our gut microbiome would produce these automatically, and we wouldn't have to take yet another pill to replace something that should naturally be there. The studies, to me, just show how powerful these compounds can be, and they would normally be generated through our microbiome. So just towards the end, and that's probably the last thing that Esther has mentioned and I have not mentioned yet, and that is metabolomics. And we really have an interest in metabolomics for a number of reasons, because that brings so nicely together the genetic background, but also, of course, what we eat and what kind of metabolites our body creates out of what we eat. And we worked a lot with the group of Alte Schutte in South Africa to look specifically, and I know this is a very crude separation between white and black South Africans, but sometimes it helps to go for these broad categories and show some differences here that then can be explored much further. And what we've done in this study is to go back to the very basics and say, if we're interested in blood pressure is determined by the vascular resistance and by the flow. And as much as we have here in our garden hose, not much flow here, but we can change it either by putting more power here into the hose and just turn on the tap, or we increase the resistance by these fancy devices, and then the flow or the pressure will be much higher in here. So for me, the vascular resistance is really, really important. And resistance is important because vessels can remodel and can get tighter, and that would increase the blood pressure. So we use, again, omics technologies here, from genomics to down here metabolomics. And the reason is really that diet, as we've seen in the very beginning, would really influence not only which kinds of proteins we synthesize, but also which kinds of metabolites are generated from these proteins in the longer term. And Alta's study here really included a number of, or a relatively large number of people. So there were Black and white South Africans that were then taken out of the large cohort that they have collected, but then extremely well matched. And so that's why the numbers here are smaller. We have used 3 different methods to look at metabolomics here. And I don't want to show you all the details, but you see very clearly that there was, throughout the 3 different methods, there was a difference between the 2 study groups in certain metabolites. And that is quite interesting because it shows you directly that these groups have either a different genetic background so that they can metabolize a standard diet in a different way, or they have different dietary habits. And in reality, it's of course a combination of both. But you see that, and these are otherwise healthy young people, you see that there's already very subtle differences. And when you look at this, what it really means, you see already in these very young 23, 24-year-old people with no cardiovascular disease that their central systolic blood pressure is slightly higher in the Black people compared to the white group here. And that could point towards some sort of increased vascular stiffness. And that is really, when you look at all the differentially expressed metabolites, many of them were involved in collagen biosynthesis. And that is, of course, the factor that drives vascular stiffening and vascular remodeling. So to bring all this together, we saw that between the Black and the white group, we have differences in metabolites. These metabolites are associated with collagen synthesis. And this already at young age, when we don't see disease yet, translates into just a tiny difference in their vascular stiffness. And that could, in the longer term, explain the higher event rate, the higher rate of cardiovascular disease in one group compared to the other group. So these studies can be quite powerful to show very early changes that then 20, 30 years later translate into clinically overt conditions. So that was the summary that I've just given you, that there was a unique metabolomic signature between these Black and white groups, and that could in the longer term translate into cardiovascular diseases. So just to finish this with one more slide, I've mentioned to you that diet can directly influence metabolites because diet contains the building bricks for our metabolism, but the diet can of course also influence what kind of proteins we can synthesize. And that's why we use a lot of proteomic efforts as well here in a study in heart failure, for example, where we generate these networks. And the multi-omic approach that Esther has mentioned is probably really the future if we ever crack the bioinformatics behind this. But of course, we would like to look at all levels, from the genetic background to how does this translate into proteins and how does it then interact with the environment to drive the metabolism. And if we then take the gut microbiome into account, I think then we've got the full story. But what is interesting is that, again, these metabolites could play a role. And what we've seen before in very early cardiovascular disease, where there is actually no disease yet, if you look at people with advanced disease who already have heart failure and you look at admissions for heart failure, you see again differences in certain metabolites between people who are and people who are not at risk. So these techniques can be really, really powerful. So let me just summarize very briefly. What I've shown you is that if you want to analyze it robustly, the gene-environment interaction, then you also need robust biostatistics, and that is something that still needs to be developed. There are tools available, but with a lot of assumptions, and that needs to be done very carefully. We know that lifestyle modifications work. We know this from our own daily personal life, but we also know this in larger populations, and I've shown you the pollution data, for example, but you also know, of course, about initiatives to reduce salt intake, for example, in South Africa, but in many other countries as well. The pollution data I've mentioned, and you see what direct effect this can have on hypertension if we just change the quality of our air. I've shown you some very simple data on the gut microbiome, because I think that's a nice way that translates environmental factors such as diet into cardiovascular diseases. And I've given you a little bit of an idea why we do not only do genetic studies or genomic studies, but go beyond this and use proteomic and metabolomic studies, because I think they would be much more directly influenced by environmental factors and give us much more of an opportunity to study these factors. And with this, I would just like to thank the British Heart Foundation, who fund most of our research, and I look forward to some discussion with you. Thanks very much.
Thank you very much for that interesting presentation. And I can see that there are a lot of questions in the question and answer chat box. But before we go on to the question and answer, I'd like to give an opportunity to Dr. Richard Dyer to make a comment about the African Research Universities Alliance.
Yeah, thank you very much, Nancy. Actually, I think I should have spoken earlier on before the start, but just by way of introduction, that this is part of the ARUA, the African Research University Alliance, non-communicable disease Center of Excellence. And what we have been talking about is one of our thematic areas is about the genomics and look at biomarkers and eventually looking at biobanking. And the work that we're doing here and what has been presented to you by Dr. Esther Maina and Professor Delis, Christian Delis, is we want to encourage participation in looking at this area in terms of the research that we do, so that we can begin to understand exactly what is ailing us, so that we can be a little bit smarter in terms of our diagnosis and management of especially of non-communicable diseases. I think the way in which COVID has come, it certainly pointed out to us that those with non-communicable diseases are a very vulnerable group. And I think one of the questions we've been asking all along is, why aren't Africans affected? And then we've come up with all kinds of reasons, basically speculation. And if we can start to get some research going in these areas, and Dr. Esther Maina is our lead in this area at the University of Nairobi, then we can begin to answer some of these questions, or at least we'll be prepared with some answers when we get through. So the Center of Excellence is a 5-university, 5-country collaboration. So we have University of Nairobi together with Ghana, Ibadan, Wits, and also Makerere. And then we have some very strong collaboration with the University of Glasgow as well. So I'd put something on our on the chat in terms of what our website is, but you can reach out to me as well. And then later on we can engage a little bit more, and it's multidisciplinary. So the first webinar was on the social sciences and COVID. This one is between clinical and the bench sciences as well. And the idea is that when we think about the patient, we have to think, as professors talked about, the environment as well. We have to think about the public health aspects. We have to think about the bench sciences that are going on and the social sciences. So thank you very much. And I think we can welcome questions at this point. Thanks.
Thank you very much, Dr. Richard Dyer. And that, I think, also answers a few of the questions that have been asked. Dr. Wambani Sidika, I guess that answers. The question she had was, is it possible to have a collaborative research project involving all the medical disciplines. And based on Dr. Aya's introduction, this is actually ongoing at the African Research Universities Alliance, and you can reach out to them and be able to collaborate with them. So there are several questions regarding the difference between our African genes and what is known. And maybe this is to Professor Christian, a question from Anto. How different is African genes variant ambiguous in genetic studies in manufacturing of antihypertensive drugs?
Yeah, I think it's a good question. And I think Esther has much more to say about this as well, but I think she said it very clearly already, and I can only confirm this. We actually don't know because there is no data available or no large-scale data available. So we know, of course, historically a few things. We know, and it's maybe a bit more of a rumor than firm knowledge in terms of hypertension, for example, that we also always say that the renin-angiotensin system is less active in African people, and that we therefore would not recommend ACE inhibitors and ARBs as the first-line treatment. But in reality, I'm not sure if it makes a big difference, and I'm not sure if this difference really goes beyond an immediate blood pressure lowering effect, or whether it also goes towards the other cardioprotective effects of these drugs. So that could be something that can be analyzed on a kind of genomic basis, on a proteomic basis quite easily, but we don't have the data. So even for these commonly used drugs, we don't really know what is the genetic reason for any differences in the action. And just imagine how difficult it will be to develop new drugs based on no knowledge. I'm not sure, Esther, do you have other thoughts on this? So my answer would be quite simple to say we don't know. We know it's different, but we don't know the details, but probably you know more.
Yeah, thanks, Prof. I think I will again agree with you. That lack of data, because science has to be standing on something, we must have some evidence. We have no evidence-based anything. And that's why I'm saying that like my speech today or my presentation was to normalize this narrative that we really need to get in there and really get ahead, because there are advances in technologies that are enabling us to actually quicken our paces. But again, as you're saying, yeah, it is what it is. But I'm hoping that this is not a narrative that will be going on for the longest while, so that many of us can just get on board, and the next sessions that we have will be more productive to answer such questions. Thank you.
Thank you very much for that. And I guess the communication to the attendees is there's a huge gap in genomics in Kenya and in Africa, and it's a field that I think as attendees and listeners to this presentation, it's something that we can take up to improve our healthcare management of the patients, even of the patients that we take care of. I'll ask this question. I'm not sure whether the answer will still be the same as the one that has been given, but I'll ask the question that has been asked by an anonymous attendee. Has human interbreeding brought about an actual significant uprise of genetic disorders and cancers here in Kenya? And what is the case, especially in communities that uphold it as a tradition? I'm still not sure whether there's information out there about that, but.
So let me go first, and then Prof will correct me, because this is a very ambiguous. People by interbreeding, I didn't quite get that, like inbreeding, or sorry, if it's inbreeding and cancer, I don't think we've seen anything like that from the work I've studied before. But again, we have not had time to even look at issues of consanguineous relationships to do with cancers. But there are other genetic problems, of course, because of linkage analysis that are actually quite geared towards linkages problem, like when you look at autozygosity mapping of diseases like Bardet-Biedl, and those have actually been seen in the Pakistani communities because of inbreeding, the Ashkenazi Jews, issues of the respiratory illness and stuff like that. But in terms of cancers, we have not looked at sort of inbreeding, so to speak. I think we're just right at the formative setting up of research, and I think asking the bigger questions about the genome, trying to understand it first, and then look to see does lifestyles, and I don't know if that is a lifestyle of inbreeding, would it have an impact? Of course, when genes that are deleterious to the system amalgamate together, subsequently they will give something that is not quite positive, but I'm not in a position to answer that conclusively. Yeah. So, Prof,
yeah,
kindly.
I'm certainly not a cancer person, so I wouldn't know much more about this. But the theory is, of course, true that, as you nicely said, if you inbreed, then you would accumulate bad genes, probably to some extent. And we know this, of course, from the cardiovascular area a lot. And you don't need to go to communities in Africa. You have this across the world, of course. And you mentioned Ashkenazi Jews. You know that there is also in Europe, there are valleys in the mountains in the Alps that have never exchanged with the rest of the world for hundreds of years. And that's also very inbred communities there. And they have indeed certain diseases that are more common. We know also in some North African countries where there is certain heart diseases, very early onset childhood atherosclerotic disease in these families. So that is possible. For cancer, I don't quite know, but I would not be surprised if the same would be the case there.
Thank you very much for the responses. Another question from Ezekiel Mecha to Dr. Miner. Why do you need to do both genomics and transcriptomics?
Okay. Sorry, finish. Or was it finished?
Yes.
Okay, so why do we need to do both genomics and transcriptomics? Genomics in its entirety encompasses even the proteins, all the genes and everything, but transcriptomics is specific for your mRNA, your messenger RNA, which carries genetic information, which subsequently goes to produce your proteins. So it is important to look at the transcript standalone in genomics, which are your proteins, your DNA. So mRNA standalone gives you a better view of how inheritance or subsequent production of function is, which is your protein. So transcriptomics is important because it enables us to follow not only the gene, but with its subsequent output, which is the protein. So, and without the mRNA, which is the transcript, the transcriptome, then we don't have function, because we need the mRNA to go to the cytoplasm and give us the protein. So personally, I'm interested in gene expression because I'm wanting to see, like, is differential expression of specific cancer genes actually a causative reason? Because you might have some genes that are actually not carrying or harboring any mutations, but because of how they are passed on, or the levels, which is where transcriptomics brings in issues like microarray analysis, real-time PCR, where you're able to, in real time, quantify. So the quantification matters of haploinsufficiency and the resulting effects of gene expression levels. And so you can talk about gene expression and disease manifestation. And you can talk about gene expression and the protein it produces. So without mRNA, you can't really talk about function, which is protein. So I think in a nutshell, that's how I would put it.
Thank you very much for that answer. We're coming to the end of the session, and even as we come to the end, I'd like to give our presenters some time to give us a few parting. Before we come to the end, you can see Dr. Omo Anzala. Professor Omo Anzala has raised his hand. If you can ask your question before we come to
Yeah, I should say thank you to Christian and Esther. This is great. My name is Omo Anzala. I'm from CAVI, Institute of Clinical Research. This is great work, and these are the areas that are also totally of interest to us. I must say that we have started looking at immune metabolism with our group from Germany. It's a totally new area to us, but we are beginning to build expertise. What I would also suggest that at this juncture, if this area is going to grow, we need to bring in postdocs as quickly as possible, because those are the guys who will really help us get this area to grow. So immune metabolism, we are looking at it from the point of view of HIV, just trying to understand how chronic immune suppression affects immune metabolism, and also how that impacts on vaccination. For example, we have been giving Ebola vaccines to HIV positive individuals and asking ourselves, do they respond or do they not respond? And if they don't, or if they do, how does the immune metabolism actually impinge on that? And we are actually looking for a postdoc, and we haven't found one. All right, we are looking for a postdoc to help us here, and we have actually not found one. So please let us collaborate on this. And then the other area we are also interested in is ARV antiretroviral metabolism and immune and genetics. We have seen that the way we metabolize ARVs as Africans could be different, and it leads to different side effects. And this could also be related to the genetics of what we are and how we metabolize these drugs and the side effects that accrue. So again, here, we want to bring in a postdoc. We don't have one. So we see a linkage here. So Esther, Christine, Richard Aya, please help us, bring this work together. And the other thing is that we've also worked with India. We've brought in a platform to begin to look at bioinformatics. We are also looking for a postdoc. We don't have one. So we cannot train masters if we don't have postdocs, because some of these things are completely new. When you talk of genome-wide association studies, we really need that. So Christian, Esther, Richard, Aya, what I'll tell you, if we are going to grow, we seriously need postdocs to begin to take up these positions and help us now train at a master's level and also at PhD level. Thank you very much.
This was the probably best job advert I've heard for a long time, but let me just comment very, very seriously on this, Omi. That was really helpful, because that's extremely topical areas, as you mentioned. And I completely skipped the whole area of infectious diseases in my presentation, but we discussed this at length when we set up the Center of Excellence, that one of the unique things that only you can do is these studies into infectious diseases that are so widespread in the population. When you mention HIV, when you mention malaria, when you mention Ebola, when you mention vaccine programs, the interaction between infectious diseases, the underlying genetic risk, and all sorts of other, especially cardiovascular and cardiometabolic diseases, and then link this obviously to the immune system, that is something so unique that nobody else can do in the world. And I'm sure you will find your postdocs, because that's exactly what is happening research-wise at the moment. So it couldn't be more topical. So I can only try and support you, at least morally here.
I will echo what Kristen has said. And the other thing is we do have quite a few PhDs coming up in Kenya who are probably in immunology, informatics standalone, but we're coming to a time of multidisciplinary as a PhD. In as much as you are an immunologist or a bioinformatician, you should be able then to come together and solve other problems by working in groups and collaborative efforts. So towards that end, the one thing that I would also advise to my fellow PIs is to actually deeply ingrain in our students that wanting to make a paradigm shift from just sorting out a specific problem, but to actually open up our minds to do a paradigm shift of how can I apply my immunology in HIV research and so on and so forth. So I think with a bit of training, a little bit of outside training, our PhD students that we're actually churning out at the moment can actually be harnessed and be well prepared to join such formidable research groups at CAVI. So yeah. So thank you, Prof.
Okay, yeah, thank you very much. And I'm glad that this is being discussed now, because we want to be able to see this area very grow. There's all issues of precision medicine, and we cannot do that if we really don't understand how our own genes impact on all these things we actually confront on a regular basis. So we are here. We are really ready to work together. Richard, you know us very well. So where we can converge, please let us know. Thanks.
Thank you very much for that. And we're glad that even the webinar sessions are bringing up issues that we should deal with as individuals who are taking care of patients and more into now doing research and into innovations. And that's a really key thing, especially now in Kenya and in Africa moving forward. We have come to the end of the sessions. A lot of the questions have actually been addressed. So we've come to the end of the session, and I'd like to give Dr. Esther Maina and Prof. Christian Dales.
Hello. Hello.
Hello.
Hello.
Hello, we can hear you.
Yeah, I had raised my hand, and kindly allow me to make a comment. Yeah, I'm Margaret Oluka. From the School of Pharmacy of the University of Nairobi. I just want to also thank Dr. Esther Maina and Dr. Christian from Glasgow for bringing us together with this very interesting area. I just want to highlight the fact that in the School of Pharmacy in University of Nairobi, we have also been interested in genetic work, and particularly pharmacogenetics, pharmacogenomics. And Esther is aware of this. We have had a few chats together. And Professor Omwanzala, actually, we examined one of my students with him who was looking at antiretroviral use in long term in patients and the changes in metabolomics in those patients. We have also collaborated with the researchers from Karolinska and Muhimbili who have worked on HIV drug metabolism, particularly efavirenz, and coming up with actually very novel data to show that efavirenz is metabolize much slower in African populations than the Caucasians and Asians because of the polymorphisms in CYP2B6. And this work was actually being used to try and modify the dosage regime for efavirenz, which is one of the leading drugs in HIV in our continent. So I just want to echo the fact that this is good, and going forward, we shall be reaching out to Professor Aya so that we work together as a team in the University of Nairobi. Actually have a student who has approached me recently that you'd like to do genomic work in hypertension. So I'll be reaching out to Esther maybe to see how we can collaborate and how this student can work with Professor Christian. So thank you very much. And I think the collaborative efforts will bear fruit in this area. Thank you.
Thank you very much, Margaret or Lucas. Sorry, I had not seen your hand being raised. Maybe I can now give the presenters an opportunity to give their parting shots. Even on the comments, I can see there are several postdocs who are interested. So also we can give probably contacts or ways in which they can reach out in case they want to be part of this great work. Thank you.
Okay, thank you, Nancy. I would just like to again emphasize that we need to normalize the narrative on African genome and issues of multidisciplinary sharing of samples and, just pushing forward the African story so that it actually becomes research for us and by us. We have a lot of goodwill from our friends in Europe and the Americas. So let us utilize these opportunities to actually train more and get more sort of like capacity building in our resources. And actually, it is really good. Sorry, I actually snuck into the question and answer, and I saw Mohammed saying that he's there. He's my former PhD student, Dr. Taha. I know you're a good guy, and you're well ready for a postdoc. He's Egyptian. So I supervised this PhD, so I just wanted to recognize him. It's a small world after all. And anyway, that is how the world should then become a collaborative effort, disciplines, and everyone to bring to the table what there is, because you have seen that Africa is way back. I would want to then in another session, as Prof. Deo has presented fantastic work on hypertension, that I should be able to put out my multi-omic results on the prostate cancer. That is the next agenda. And so that way, and if there's one of me and all of us doing that, we will make leaps and bounds. So again, thank you so much. And thank you, Prof. Deo, for, it has been good. It actually worked well. Someone would have thought we planned together, that I lay the foundation and you put in some application work. So it has been really good, and it has been interesting learning this. Thank you so much, Dr. Richard Aya, and thank you, Arwa, for creating this fora where you bring in the African scientists for all of us to, because I remember the first time I spoke with you, Daktari, I was complaining that it has been 10 years and no one listens to me when I keep saying that we need to look at the Kenyan genome standalone. And with such fora and many more to come, working together, the bench scientist and the clinician, therein lies our success. So thank you very much, everyone. Thank you for the opportunity to present in this session. And I look forward to more fruitful discussions in the future. I've put my email address on the question and answer section, and I look forward to hearing from people. Thank you very much, Nancy.
Okay, and really not much to add from me. So same, of course, if there's any drive to collaborate or any wish to collaborate, I'm happy to answer emails, no problem. One thing I should say, however, I mean, these cancer guys have, of course, an easy life. because when they do multi-omics, it's not a big problem because they always have tissue. So if you do prostate cancer and look at prostate cancer cells, you can do all sorts of fancy things, and that's to some extent easier than if you look at cardiovascular diseases or diabetes, where we would not be able to do this in so much depth. So that would be probably an interesting discussion point, and I really like your idea to bring these multi-omics ideas together at some point and see what kind of opportunities you have in the cancer area and what kind of challenges we would have with other more common diseases. So that would be really interesting. And otherwise, I can just echo what else you have said. I think what is important is not just to say, well, unfortunately, we are 10 years behind. It's also a great opportunity. And what I said before is that there are so many conditions that you can study that are just so many opportunities. So let's not just say, unfortunately, we are so many years behind. It's also great opportunities. And I think the drive is there at the moment to address this. So thanks very much for the opportunity. It was great to see Richard again and many others. So thanks very much.
Thank you very much for the presentation and for the education in terms of genomics and the research gap that is existent. And a lot of people have actually expressed interest in issues of genomics and health. So kindly, we've come to the end of this session. Tomorrow we'll be having a session and we'll be doing all matters thyroid. And we'll be having a surgeon and a physician discussing everything to do with thyroid disease, thyroid awareness, signs, symptoms, just everything thyroid, in commemoration of January being the Thyroid Disease Awareness Month. So kindly tune in with us tomorrow from 2 to 3:30 p.m. For pharmacists, as I had initially stated earlier, we are unable to put the webinars on the Pharmacy and Wholesale Board portal. Therefore, the system that is being used for reporting is the self-reporting icon. So kindly click on the self-reporting icon to make your CPD claims. It will request you for some details and evidence of CPD attendance, and the evidence we will send that to all your emails. So please be sure to state the title of the webinar session that you're claiming. And in case of any issues, kindly email us at cpdknetpharmacy@gmail.com or knetcpd@gmail.com, and we'll sort you out. So I know there have been a lot of queries about how to claim CPD points. And right now, as I'm saying, we are not able to put this on the PPB portal. It's something we are working on, but in the meantime, you can claim your CPD points through self-reporting. On Friday, we'll be having the session on experiences in managing COVID between 10 and 12 p.m., 10 a.m. to 12 p.m. And then we'll also be having another session from 2 to 3:30, where we'll be looking at the overview of cervical cancer screening. It's also cervical cancer awareness, and we'll be looking into that. Thank you so much to all our attendees who attended, for listening in, for staying in up until the end of the session. And thank you to the Research and Programs Department on the background who are facilitating this webinar. Have a nice afternoon.
Thank you.
Okay, bye-bye, everybody.
Thank you. Bye.
Bye-bye, and thank you.